Hi, hello everyone. My name is Natasha Adriana binti Faizul.
Hello, my name is Muhammad Azrizan bin Anwar.
Hi everyone, I'm Siti Afrina.
Hi, my name is Darren Arvind Singh.
Hello, we are from CE1 Group 4 and this is our video presentation for our integrated design project. The title for our project is Biogas Production from Anaerobically Treated Foam in a Two-Stage CSTR and the Purification of Biogas through Adsorption and Absorption Processes. These are the objectives for this part. The feed for our reactor system is the palm oil mill effluent or POM. It is a viscous brown fluid that has suspended solid with a pH of around 4.5. It has a high concentration of grease, free fat and oil, along with crude oil solids and dissolved oil. And so, converting the organic matters inside POM to biogas is a viable method to treat POM. Biogas is produced through anaerobic digestion. Table 1 shows the average composition of POM. Anaerobic digestion is the breaking down of organic matter such as carbohydrates, proteins, and lipids to produce biogas and occurs in the absence of oxygen. There are 4 stages during anaerobic digestion, which are hydrolysis, acidogenesis, acetogenesis, and methanogenesis. This is the reaction during hydrolysis, and this is the alphabetical representation of the equation. Based on our research, the reaction rate constant K1 is 0.162 per day. It is assumed that water, which is B, is fed into the CSTR in excess. The microorganism that is present here is the hydrolytic bacteria. Moving on to the acidogenesis stage, the K2 is 0.08 per hour. It is assumed that C is reacted fully in this reaction. The second reaction does not occur as water is present in excess. The microorganism that is present here is the acidogenic fermentative bacteria. For the acetogenesis, the K3 is 0.4 per day. The first reaction does not occur as G is not generated during acidogenesis. As C is fully reacted during acidogenesis, the second reaction also does not occur. E is assumed to be fully reacted as it is reacted with excess water. The microorganism that is present here is the acetogenesis bacteria. And as for the methanogenesis, the K4 is 0.06 per hour. The second and third reaction will not occur, and the microorganism that is present here is the carbon dioxide reducing bacteria and the acetylclastic methanogens. Moving on to the selection of reactor, we have chosen to utilize the continuous stirred reactor or CSTR as it is a good reactor for the treatment of substrate with high suspended solids and FOG content. It provides good contact between biomass and wastewater, which increases the production of biogas. The cost is also low and it is easy to operate. This is the proposed process flow diagram for our reactor system. CSTR1 and 2 both operates at 38 degrees Celsius and 1 atm. The pH value for CSTR1 is 6 with the retention time of 12 days, while the pH value for CSTR2 is 7 and the retention time is 20 days. After performing the stoichiometric table and also through the 5 steps method that we have learned in chapter 4, it is found out that the volume for CSTR1 is 2300 meter cube and the volume of CSTR2 is 369.3 meter cube. In order to maximize the yield and selectivity of biogas and minimize the side reaction, a suitable reaction temperature must be chosen, adding alkali with molarity of 1 molar to the POM, which is also known as alkali pretreatment, and finally, the excess sludge from effluent is recycled to the feed.
Now, we are moving on to the process and modeling simulation part. These are the objectives for CEB 2053 process modeling and simulation. For this case study, the students need to develop a modeling equation that enable the determination of species concentration and mass balance inside the suitable reactor in order to maximize the yield of biogas and produce biogas that meets the specification. In order to develop a modeling equation, there is 3 major parts that need to be included. The first part is the mass balance of the system. In this case, we've constructed the mass balance for CSTR. Next is the composition balance. in which every component should be taken into account. In this case, we've constructed the component balance for component A, B, C, and E, H, and lastly F. And the last part is the energy balance, in which we need to construct the energy balance for all the system that is involved, which includes the CSTR1 and CSTR2. Based on the model equation constructed, we can find the concentration of the component. For this case, for component A, based on manual calculation, we find that the concentration of A in CSTR1 is 0.496 mol per dm cube. However, based on Aspen HYSYS result, the concentration of A in CSTR1 is 0.5177 mol per dm cube. Using the following formula, we can find the deviation percentage between Aspen HYSYS and manual calculation value. The following are the deviation percentage in CSTR1. And here are the deviation percentage in CSTR2. It is clearly can be seen that there are deviation errors in all the values when comparing HYSYS simulation and manual calculation. Besides, the manual calculation is made according to the model equation and many assumptions has been made to simplify the equation, which has caused deviation error. All in all, manual calculation is mainly for reference in modeling reactors. However, real-life processes must be taken into consideration, as such assumptions made to be simplified the model equation are hard to be done realistically.
So for the process simulation by using HYSYS, fluid package used was BWRS. The feed conditions are obtained from the reaction engineering part. The feed is operating at 38 degrees Celsius with 1 atmospheric pressure. Since the reactor should be at the capacity of 30,000 tons per year, the feed is fed at 3,424.66 kg per hour into the first reactor. So in the first reactor, there are two stages take place, which are hydrolysis process and acidogenesis process. Meanwhile, in CSTR2, it involves acidogenesis and methanogenesis. Figure 1 shows the process flow diagram obtained from the simulation by using HYSYS. Meanwhile, in Figure 2, shows the components composition leaving CSTR2, which will be purified later in the separation process. Sensitivity analysis is done to observe the effect of different pressure, temperature, and flow rate on the performance of the reactors. In this analysis, one parameter is manipulated while the other two parameters are kept constant. So in graph 1, we can see that as the pressure increases, the conversion inside of the reactors increases as well. Meanwhile, in graph 2, we can see that as the temperature increases, it will affect the conversion and the conversion will be decreasing. It shows that the conversion is actually inversely proportional with temperature. And lastly, in graph 3, it shows the conversion versus flow rate graph. As the flow rate increases, the conversion inside the reactor will decrease.
Hello, I am Darren Arvin and I will be explaining on Chapter 4, which is the design of downstream separation process system for the removal of contaminants from the raw biogas, which is highly related to our course separation process 2. The objectives of Chapter 4 are listed on the slide. The first objective is to design downstream separation processes in order to remove contaminants in raw biogas. The second objective is to discuss the process of each separation unit involved in detail. The third objective is to discuss the safety measures that should be taken, cost, and also the maintenance of separation units. The fourth objective is to discuss the challenges that will be faced and also solutions to the challenges. And the fifth and the last one is to discuss strategies to maximize production. Figure 4.1 shows the process flow diagram of the plant. The plant is divided into three main parts, which is the adsorption unit for H2O removal, the absorption and adsorption unit for H2S removal, and also the pressure swing adsorption for CO2 removal. In part 1, the process is adsorption, and the purpose of this is to remove H2O from biogas stream, which is water. The type of adsorption that will be used is fixed bed adsorption, and the type of adsorbent is silica gel. By referring to the graph in figure 4.3, the targeted removal efficiency of H2O is between 95 to 99%. The next part, which is part 2, is the absorption and adsorption unit for the removal of H2S, which is hydrogen sulfide. The purpose of this unit is to, of course, remove H2S from the biogas stream. The type of absorption that will be used is absorption using iron as absorbent, while the type of adsorption that will be used is fixed bed adsorption. This adsorption process is also used to remove hydrogen sulfide from the biogas stream as well, and the type of adsorbent used is activated carbon. As for the efficiency of the absorption unit that will be removing H2S, the graph on figure 4.6 will be used as reference. The percentage removal was calculated and the targeted efficiency is 80% removal of H2S. As for the efficiency of the adsorption unit that will be used to remove H2S, eucalyptus activated carbon was taken as the basis, and the adsorption capacity that is targeted is 600 to 700 milligrams hydrogen sulfide per grams of adsorbent. In part 3, pressure swing adsorption will be used to remove CO2 from the biogas stream, and the type of adsorbent that will be used is zeolite. As for the efficiency, the figure 4.8 will be used as the basis, and from the basis, the energy efficiency that will be targeted is 0.3 kilowatt hour per meter cube, while the CH4 slippage will be targeted so that it doesn't exceed 2%. Safety measures will be taken by referring to the safety data sheets of materials. As for the cost, after doing some rough estimation and calculation, the total cost that is estimated is around 500,000 ringgit. And as for the maintenance of optimum conditions for each separation unit will be done so that they can work well. And with that, we have come to an